I can't wait. Yeah, it just seems so far into the future still. So we may be like this for another 6 months or so until, March or April. I don't know. I guess we'll have to wait and see. Okay, so let's go ahead and get going. So I am Lori McMahon. I'm Dean of the Graduate School. I welcome all of you here this evening for our next installment of Discoveries in the Making. And so those of you who have participated prior to COVID know that we typically have these events in a science-inspired bar, the Lumbar, and the owner of the Lumbar specifically installed equipment so that we could be in his place of business talking about science and sharing the knowledge that we are generating every day at UAB. And that's what we do in all disciplines. We're generating new knowledge. We're working really hard in many disciplines. And I feel really strongly, and all of you have heard me share this before, that it's our duty, it's our responsibility to share that knowledge with others, and others that are outside our discipline. So it could be other colleagues at UAB who are not familiar with our discipline, but also just non-experts, people who are not in research, who are not in academics or academia, and just want to learn more. And so that's why we're here. That's why we developed Discoveries in the Making. And so it's a great opportunity to have a discussion and to ask questions. So I'm sure our speaker tonight, Melissa Walker, she will entertain every question that you want to ask her. Typically, we allow questions during the presentation itself. It's a little bit different feel on Zoom. It's not so easy to do that, but I would encourage you to unmute yourself, speak up, ask Melissa a question when that question pops into your mind, or raise your hand. You can use the reaction button if you would like. And just let's have a really great discussion. So Melissa's going to talk to us tonight about the evolution of phage therapy. So Melissa, welcome and thank you for participating tonight. Can't wait to hear what you have to share.
Well, thank you so much. I am really excited and thrilled to be here to talk to everyone about something that I'm so passionate about. I love phages. I love working with phages, and I really enjoy the dynamic challenge that comes with trying to find applications for phages as solution to real-world problems. So tonight, what we're going to do is just take a look at some of the history of phage therapy. Now, we're going to talk about its beginnings, but also some of the scandal surrounding how it started. And then we're going to talk about the current state or use of phage therapy before we move on to talking about some of the challenges inherent in producing therapeutic phage, and then how my research answers some of that, particularly from the perspective of phage evolution. Now, as I move from topic to topic, what I'm going to be doing is just defining some key terms. And I want us all to be on the same page so that, we all kind of know what it is I'm saying and how I'm using those terms. But also, as we transition from topic to topic, I will also be stopping to give people a chance to ask questions. So I don't have my little chat open, but, just in case, know that I love questions. You're free to interrupt and ask a question as it comes up, but do know that if you feel like it's not the time, there will also be just a couple little breaks that people have to actually ask those questions. All right, now, if we're all ready to get started, it's definition time. And the first thing that I want to define for people tonight is the term phage. Now, when I use the term phage, what I'm doing is using an abbreviated term that refers to a virus that infects a type of microorganism. From the perspective of my work, I usually work with bacteriophages. So these are viruses that infect bacteria. Another term I'll be using very frequently tonight is the term phage therapy. When I use the term phage therapy, what I'm referring to is the use or application, therapeutic or medicinal, of phages to treat some type of disease. Now, from the perspective of my research, the applications for my research are really within the realm of medicine and biomedicine, but that's not the only area that phages can or have been used. Phages have been used to treat different types of animal diseases as well as different types of plant diseases in agriculture. So now that we're armed with those definitions, let's go ahead and talk discovery and just a little bit of scandal. All right, so back in the 1800s, tobacco was a huge staple crop that brought in a lot of money. And the farmers started to complain because they noticed that the leaves on their plants were wilting, they were turning yellow, and they were starting to die off. Now, this was really bad for business, and it impacted everyone from the little farmer all the way up to big companies. So one particular researcher by the name of Dmitry Iwanovsky decided that he was going to try to isolate what was causing that disease. So he took some of that diseased plant leaf material and he crushed it up, and then he filtered a little bit of liquid through that plant material through what they call a porcelain filter. Now, porcelain filters were supposed to be filters that could filter out every known organism known to science at that time. So that included bacteria up to even larger things like algae and stuff like that. All right, so now he's filtered that liquid and he takes some of that liquid and he applies it to the leaf of a healthy plant. And what he finds is that he's able to transmit that disease from a sick plant to a healthy plant. So now he knows that whatever was in the tobacco leaf was filterable, it was tiny, and it was also transmissible. So let's fast forward about 6 years to 1898, and we see another researcher by the name of Martinez Bayerent. Now, Martinez really wants to try to characterize that filterable agent a lot better. And the result of his work is actually the first definition that we have for a virus. And it was very simple. It was essentially just a small particle or molecule that once inside the cell of another organism became reproductive. Now, this was really novel for science at that time, and they still had no clue what it was that was doing this. They didn't know if it was a bacteria or some like new type of biological entity they'd never seen before. They didn't really know. So now let's talk a little bit about scandal, okay? Now we're gonna start talking about scandal with a researcher by the name of Felix de Herel. Now, Felix works with children in the children's ward who are dying of dysentery. Dysentery is like explosive diarrhea, and it's caused by a bacteria. So he's growing this bacteria on a plate, and what he notices are that there are these clear glassy plaques, what he thinks are zones of death essentially, where something has affected the bacteria and the bacteria have died off. Now, when I use the term plaque, you can picture in your mind sitting in like your living room and you look at the wall of your house, and most of that wall you can't see through, but then you have a window and that window is clear, you can see through it. The same is true when bacteria grow on a plate. They make the plate completely opaque. And so what he noticed was like a window in the plate of that bacteria. So he decided he was going to try to investigate just a little bit further. So he isolated that plaque and he did the same thing that Iwanovsky had done. He put it on a porcelain filter, he filtered some liquid through it, and when he got done, he took some of that liquid and he put it in a fresh culture of bacteria. And lo and behold, what happens? The bacteria dies off. So then he thinks to himself, I wonder if I can actually use this to try and treat a patient. So he goes through the process of purifying that phage liquid or that liquid so that it's safe for people to actually take. And then he administers it to just a couple select number of patients. And surprisingly enough, they recover, completely recover without any adverse issues. And so now he's really super excited because at this time, as a backdrop against the research he's doing, they are really dedicating a lot of resources to try and figure out what the underlying foundation for like the immune system in the human body is. And he thinks all of a sudden, I wonder if it's this phage. Now, he publishes his results and he publishes his hypothesis and almost immediately draws a whole bunch of fire from the scientific community. And one man in particular, this man, Dr. Jules Bordet. Now, Dr. Jules Bordet is a Nobel Prize winner who's received his Nobel Prize for his research in antibody-based human immunity. So you can automatically see like what the issue or dispute is going to be between these two guys. In one corner, you have Felix, who is not very personable, doesn't have institutional backing, doesn't have a Nobel Prize, who's like, go phages. And in the other corner, we have Jules, who is a Nobel Prize winner, very charismatic, does have institutional backing, who is like, it's all antibodies. So this dispute actually lasts between the two scientists for like the next 10 to 12 years, where Jules makes it his personal mission to undermine Felix's reputation in the scientific community. And he does a pretty good job. So it's hard for Felix to try and find anybody to really listen to his theories about these phage and what it is they do and how it is they work. So then we finally see Felix kind of reaching the end of his tether, and he's like, you know what, forget it. This is even more scandalous what he does. He quits his professorship at Yale, and he starts a phage therapy center halfway around the globe in Tbilisi, Georgia. Now I say Tbilisi, Georgia now, but at the time, this was the Soviet Republic of Georgia. So he has essentially left an allied democratic nation and like went to a communist country. Now, this is really bad for phage therapy because of the fact that it starts to insinuate or embed phage therapy into the fabric of both communism and Nazi Germany. And that's just like a couple of the nails in the coffin. There are a couple more. The other one is really the development of antibiotics. So in 1928, while this whole dispute between Felix and Jules is going on, another researcher by the name of Alexander Fleming discovers antibiotics. He discovers penicillin. And so he and some other researchers and a couple of companies spend the next decade really developing this into a commercialized medication. And what we see is that this medication is heavily utilized by the Allied forces during World War II. It's also at that time that we start to see the establishment of what we now recognize as big pharma companies. And they discovered that making these medications large scale for commercialization was actually very lucrative. In addition to that, there are some benefits at this point in time, and even currently today, of antibiotics over phage therapies. So antibiotics are relatively easy to compound. They can be largely manufactured on a commercial scale. They can be packaged and transported without too many considerations for breakdown in the medication itself. And at the time that this went on, they could be used to treat any number of diseases caused by bacteria. So you could give penicillin at this time for anything from like a small wound infection on the foot to like an ear infection to a respiratory infection. Now, the advent of multi-drug resistance in bacteria has really, I think, opened the West up, or Western medicine, so to speak, to reconsider phage therapy. So it's going through what they consider to be like a renaissance right now. All right, so just a quick summary here is that we've gone through some of the scandalous history, and we see that because of the war and the way that where and when that phage therapy was beginning to be developed, phage therapy was also used as a main treatment modality by the Central Forces during World War II. So you see that it's really associated heavily with like communism and with Nazi Germany. And so a lot of scientists didn't really want a lot to do with that at the time. And then also there are just these advantages that antibiotics have over phage therapy that kind of make it more lucrative as well. And so before we move on, I just want to ask, are there any questions? And I can't see my little chat box. Let me check the chat box. No, no questions. Okay. All right. So now it's definition time again. And there's just a couple things that I want to point out before we move into just a short recap of like the current state or use of phage therapy. And the first one is a phage cocktail. So a phage cocktail is essentially a distilled liquid that has different types of phages in it. Just like there are different types of bacteria, there are also different types of phages. And sometimes when they make these cocktails, they want to include a number of different types of phages that can treat different types of bacteria. And the other one that I want to mention is just compassionate use. Now, compassionate use is a term really used in Western medicine. And what happens is that traditional medical treatment for a patient has been exhausted and there's really no other options. So compassionate use refers to the use of experimental treatments that haven't been approved by the FDA to actually treat some type of medical condition. And phage therapy very much so falls under that heading here in Western medicine currently. And so the current state, what we find here is that there are really two options for receiving phage therapies as a treatment. The first is pre-prepared phage cocktails. Now, they don't really have these here in the United States on a widespread scale like they do over in Eastern Europe. The countries of Poland, Georgia, and Russia, as well as France, have continued, and India too, have continued to use phage therapies and have developed them over the last 100 years or so. And so they have a lot of these pre-prepared phage cocktails that you can go right online and order. However, sometimes the infection is so severe that they don't want to just apply a general or non-specific phage cocktail to it. So then what we see are these personalized phage cocktails. Now, what happens is that you can take a swab from the patient's wound, or if the infection is in the blood, you can take a little bit of blood, and then you send that sample overseas to either the Eliava Phage Institute or the Phage Therapy Center that was started by Dr. Harel. And what they do is they identify the bacteria that's in there, and then they try different phages to see whether or not the phages will kill that bacteria off. Once they find phages that will actually kill the bacteria or the multiple bacteria that are in the wound, then they will go ahead and send that through to the patient for them to take. And what you see here is essentially a plate of bacteria, what we call a bacterial lawn, and these little circles that are on this plate are the plaques. Those are like the clear, glassy clear areas that D. Harel or Felix reported and were also reported by Twart. So currently, I kind of covered a lot of the stuff in this slide already, is that current use in medicine really is overseas. So you can get phage therapy pretty much commercially or even over the counter just by contacting the phage therapy centers in Europe. It's been practiced there for 100 years or better by now, and so they're pretty good at it. They have an established pipeline for how they produce therapeutic phage. Some of the drawbacks, though, are the fact that a lot of times Eastern medicine and Western medicine kind of disagreed until more recently about how to perform different types of medical experiments. And so they didn't really use a lot of like the double-blind placebo type experiments that we use here as part of our scientific method in medical testing. And so a lot of the results that we find from European trials of phage therapy are what they call anecdotal. They gave the phage therapy and then they just observed like the patient got better or they didn't get better or some spectrum in between. And so in an effort to try and really improve the way that those are tested, we're really trying to make improvements in how we test those phages by using these double-blind controlled experiments. In the United States, though, as I said, it's only by compassionate use. And then we also see that there is IPATH. So a couple years ago, back in 2016, there was a woman by the name of Stephanie Strathy, whose husband, Tom, acquired a multi-drug resistant strain of Acinetobacter baumannii. I can't ever pronounce that. And they tried everything that Western medicine had to offer, and he was just dying. And so she was finally able to get approval to use phage therapy as kind of under that compassionate use heading. And when he was administered that, he fully recovered, which was just amazing. But because she's also a professor at UC San Diego, she was very vocal about what had happened and was really good at using social media. And so people found out about this and they just started to email her. There were people all over the country who were just pleading for help and trying to get some of this alternative therapy to help their loved one. And as a result of that, we now have the Center for Innovative Phage Application and Therapeutics at UC San Diego. And we are actively pursuing a more integrated approach using phage therapies for some of these multi-drug resistant infections. All right, so that was a whole bunch. Any questions?
I have a question.
Okay.
So how's phage therapy administered? I mean, can you, like for the wound, you were mentioning a wound. So can you do it topically, systemically? How does that work?
That's a great question. And phage therapy can be given in any number of ways. So we can administer it orally. Some of the Phage Therapy Center in Georgia has some different wound dressings that have phage infused into the actual dressing that you can apply to a wound. But we've also tried just trying to apply the phage onto it like a powdered phage. You can crystallize phage and then apply it that way. Depending on how it's prepared, it can also be given intravenously, so directly into the bloodstream to help treat infections in areas that are hard to reach. So there's actually a number of different routes that you can administer phage therapy through.
So, why doesn't the person's immune system attack the phage?
Sometimes it does.
Okay.
Sometimes it does.
I mean, it makes sense to me that our immune system would see it as foreign and make antibodies to it and cause, you know, a big inflammatory reaction.
It can be. So there are some types of phages that are known to do that. And really and truthfully, that's where a lot of like the animal testing comes in before you move on to using something as a phage therapy in humans. You really do want to see if it triggers any kind of reaction, systemic reaction, because that is really hard to treat. And in patients who are already compromised,
right,
who are very, very sick, that can be very deadly.
Right, right. Okay, my last question is, what's the time frame of phage therapy compared to antibiotics? You know, how long does it take? Is it slower? Because I mean, doesn't the phage have to replicate?
Well, that's one of the nice things about phage is that they are self-replicating. So
Yeah.
as long as the bacteria is present, the
Right.
phage will continue to replicate. I don't know that I can answer that exactly though, how long it takes, because a lot of it depends on route of administration. So when you give a medication, regardless of whether it's a phage or some other type of medication, there is going to be a difference in how quick the medication is dispersed into the bloodstream, how fast it's cycled out either by the liver or the kidneys, and then how long like it actually stays in your system. And a lot of that has to do with the specific medication and also the route. So things that are given orally have to go through what they call a first pass effect. Right. So it's actually processed by the liver before it's distributed to the bloodstream. But then you can do things orally where you can give it sublingually, where it's more quickly absorbed. You can also give things IV, which means that they don't necessarily have to go through that first pass effect that the liver does. And then depending on the half-life, then that will affect like how fast it stays in the bloodstream.
Right. I guess I was just thinking about mechanism of action. You know, so the mechanism of action of killing the bacteria, you know, what the phage is going to do versus penicillin, the mechanism is going to be different. And so I was just wondering, you know.
Oh, yes. Well, I see. So, you know, phage actually reproduce very quickly. And well, let me take that back. Phage have a range of length of time that it takes them to actually reproduce. Some phage reproduce really quickly. So you can put phage into a liquid culture and then have it attached to the bacterial cell and reproduce and then kill that bacteria cell in as little as like maybe 15 to 30 minutes.
Oh,
And
wow.
there are also phage, yes, that then work more slowly. It takes them a little bit longer to get where they're going, takes a little longer for their DNA to get like the machinery going, and for them to make the different types of like the small baby viruses inside of the bacteria cell before it actually kills the cell.
Yeah, great,
So,
thanks.
okay. Yeah, thank you for the questions.
Yeah.
Great questions.
Hey, Melissa, you have one in the comments as well. A person asks if there are any short or long-term potential harm, if there is any potential harm from phage cocktails exposure.
That is a really good question. To this point, what they have found, as long as we screen phages well before we administer them therapeutically, there seems to be a minimum effect of the phage itself. What we see more often are side effects of the bacteria dying. So gram-negative bacteria are bacteria that have like 2 cell walls, and when a phage infects that type of bacteria and it kills the bacteria, what is left over are like these little fragments of that cell wall. And those fragments are what we call endotoxin. Those endotoxins can trigger a really bad inflammatory and autoimmune response in patients. So we have to be very careful about how we administer those. Also, phages have genes that produce endotoxins. So the fragments of those bacterial membranes that cause issues, phages produce sometimes compounds that resemble that. And those can also cause complications. So it's not really so much like the phage itself is causing the issue as like the mass murder of the bacterial cells. And so that's really what you have to kind of watch out for. And that's mostly with gram-negative bacteria. Did that answer your question? Hopefully. Yes. I can't see the...
No response, but I thought it was a good one. We also have, are we considering using phage therapy to treat MRSA in the US?
Yes, as a matter of fact, they do consider using that. So we have a number of different types of phages, phage cocktails that they use to treat MRSA. And it's a little nicer because Staph aureus is a gram-positive bacteria. So it seems like because it has a slightly different configuration in the actual membrane of the bacteria, when that bacteria breaks down, there isn't quite as much of an issue with the byproducts of that. The body seems to be pretty well equipped to clear that out. But one of the things we have to be careful with is the fact that MRSA has some, like the bacteria itself has some defenses. And I'll talk a little bit about one of the challenges to developing therapeutic phage therapies is like the bacterial resistance. So some bacteria are better at defending themselves than others, which then forces us to have to find different phages to try and treat, you know, similar infections.
Great, thank you.
Okay, well, yeah, let's push on. I love these questions. Great questions, everyone. All right, and now what we're doing is we're gonna go and it is definitely definition time. So the first thing that I wanna just define for us is host range. When I talk about host range, I am talking about the different types of bacteria that a virus or a phage will infect. Here we can see that this particular phage, and this is just an example, has what we call a limited host range. It will infect only one strain of bacteria. Sometimes it will infect, like phages can infect more than one strain of the same bacteria. This is another example. I work with both of these strains of Pseudomonas, both PA01 and PA14. Okay, but then what we're really looking for is a phage that has a really broad range of bacteria that it infects. So here you can see that this particular phage, for example, infects E. coli and Acinetobacter as well as Pseudomonas. Now, these are the ones that we like to be able to work with because of the fact that they're already equipped to infect or kill off basically multiple types of bacteria. And a lot of the times when we're dealing with sick patients, what we're dealing with is not a single bacteria that's causing an infection. A lot of times there's a single bacteria that really dominates the community of that infection, but there are multiple types of bacteria growing in that infection. Okay. And the next is just the concept of amplification. Now, this is where we try and grow phage. Now, phage don't really grow. They need a host cell, like a bacteria, to reproduce. But what we typically do to try and produce a large amount of viruses is to take a small amount of bacteria and then a small amount of viruses, and then we combine them into one liquid, and then we just let nature take its course. Like the viruses infect the bacteria, there's mass destruction, and then we get a lot of viruses as a result. And that's just called the process of amplification. But then there are two things I want us to also know about viruses, and that is their life cycle. So the first is this lytic life cycle. Now, viruses have two life cycles, and the lytic cycle is like the death cycle. So there's a virus that goes through and it attaches itself to a bacterial cell. And what's going to happen is it's going to interject its genome into that bacteria cell, and it's going to commandeer that bacterial cell and its machinery, and it's going to force that bacterial cell to make all the parts that it needs to make a whole bunch of baby viruses, okay? And once all those baby viruses are put together, then it reaches what they call like the burst point, the burst size of that bacterial cell. And then it's going to release an enzyme called an endolysin, which actually breaks the cell wall of the bacteria down and releases all of the little baby viruses. And this is just straight like mass murder of bacteria everywhere. But there's also what they call a lysogenic life cycle. Now this is a little nicer. This is where like the virus pretends to play nice for a little while. You have a virus that attaches itself to a bacterial cell. It injects its DNA. The DNA travels or makes its way to the host genome. And once it's there, it actually inserts itself into the host genome. And then it just kind of hides out there until and then the bacteria cell, it will reproduce and basically just pass on this virus DNA through its subsequent generations. And usually what triggers that to become active is some kind of stress event that the bacteria undergoes, whether it's a lack of some type of nutrient or too much exposure to the sun or something like that. Then the virus becomes active and it enters what they call the lytic cycle. Now, I want you to be familiar with that just because one of the challenges that we really find in trying to produce therapeutic phage is found in ensuring that we're getting a virus that only has a lytic life cycle. So once again, it's just looking at the pipeline for producing therapeutic phage. Our reservoirs are rather limited for where we can go and actually try to find phage that will infect the different types of pathogenic bacteria that cause issues in human health and disease. So typically you can go to a wastewater treatment plant and get some really stinky samples, and you'll find some viruses in there that will infect like Pseudomonas, maybe E. coli, maybe even Acinetobacter. But if you want to try something that infects MRSA, you have to go to like the pig fecal waste repository. I don't even know where to find those, and it sounds horrendous, but that has been, in the literature at least, the most abundant source of like staph viruses that they have found. In addition to that, once you identify a new phage or a new virus, you have to go through an entire process of characterization. You have to screen it for the life cycle. You have to determine its host range. You also have to determine like its burst size. And burst size is directly related to like how much of an initial dose you want to be able to give somebody if you're trying to treat a particular type of disease. And then there's also the physiology, like what does it actually look like? I think for the FDA to ever approve it, we have to at least have an EM photo. You'll see here that I have an EM photo of one of the viruses that I've recently started to kind of look at and work with. Another one of the challenges, though, is really in bacterial resistance. So bacteria do have some built-in defense systems that help them to fight off phage infection. And if we find that a particular phage is administered that is ineffective, then what has happened is probably that the bacteria has a way to just kind of shut it down. Another thing is endotoxins. So we talked a little bit about endotoxins, so I won't say too much about those. And I've also mentioned multibacterial infections. Usually it's not just one bacteria that's infecting a person, it's multiple. But then we also have a great need for different types of innovative delivery vehicles. So different ways to just try to get phage where they need to be. And again, this touches on what you were saying, Dr. McMahon, in like the route of administration. Like when you give patients something orally, there is only a limited number of phage that are going to make it past the hydrochloric acid in somebody's stomach, right? And then when you give it like over transdermally, where you do it through the skin, only so much is going to be absorbed. So we're constantly looking for ways to directly and more efficiently deliver phage to where it is they need to be. There are also some areas of the body that are really hard to reach. So say that you have a patient who just had a hip replacement. You know, it's really hard to try and deliver medication to that wound because of the fact that there's very little in the way of vascularization around that actual hip joint. So that means that the blood flow around that hip joint is not as, it doesn't cover as much of the area as you would like. And so it's really hard sometimes to get medication where it needs to go. So my research in particular looks at addressing some of these challenges. And the first challenge I want to mention here is some of the innovative delivery. So one aspect of my research, and it's multifaceted at this point, is to focus on a collaboration that I have with the material science department and the orthopedic department, where I do some different types of material testing to see how well attaching phage goes to certain different materials, in addition to then testing how well those materials are at trying to break down like a biofilm of bacteria. I also continue to screen for new therapeutic phage because I am a big believer in there's never enough phage to go around. And so we constantly want to be adding to the ranks of the therapeutic phage that we have to select for treatment. And in addition to that, this is one of the aspects of my research where I get to really educate our undergrads. I'm currently training undergrads to do some of this work. So I am simultaneously working with them to try and identify novel and new phages. We can publish that, and that's a great experience for them. It really broadens their horizons, but then it also helps to equip like the future generations of potential phage researchers. I'm really hoping to get a couple like really hooked on it. And then it also is a source of new phages. And then finally, there's the production of new therapeutic phage in a lengthy pipeline that I'm trying to address through phage evolution. Now this I'm not going to talk too much about because we're really hoping that it will be a methods paper and it's a little proprietary, but this would really use characterized phages that we would then take through a system of evolutionary steps. Like we would expose it to some new environments that would cause some mutation, and in causing those mutations, then what we would do is try to broaden the host range of those particular phages. And so that's really kind of what I'm working on in response to all of this, because it's just an absolutely fascinating field. And real world applications for that kind of science is that we would be able to use it in all different kinds of areas. You know, evolving phage to be specific for something has applications in veterinary medicine, like I talked about, even in agriculture, aquaculture, where we have, so we can hear or we can read the paper and hear about like the cyanobacterial outbreaks. This could be something that applies to that. And then of course in human health and disease treatment, which is just really important. And then that's it. I want to thank everybody for hanging in there, for listening, for all the wonderful questions. And I just want to acknowledge all of my lab mates, my advisor and my committee, and then also Dr. Purcell, who is a researcher from France who gave me some viruses to work with, Dr. Bill Benjamin from UAB Hospital in pathology, who was so wonderful about giving me some awesome Staph aureus, and then just everyone who attended tonight.
That was fantastic, Melissa. I'm sure everyone, Melissa would entertain more questions, so please ask.
Yes, I love questions, especially hard questions.
So Sarah, do you want to just unmute yourself and ask?
Sure, yeah, I was just wondering how many phage there are.
Right now, the estimate is about 10 to the 31st in a single drop of water. So phages are by far the most abundant biological entity on Earth. And we have found that they're very important in the ocean biogeochemical cycles and providing oxygen for our global ecosystem. And it may be that as our research into both our own personal microbiomes, we discover too that our virosphere, or the viruses or phages that live in the bacteria on our own body, are just as important to our own health as well.
So it's pretty amazing. So these things are everywhere.
They really are. They have been found in every single habitat on Earth where bacteria exist, even to go so far as the ice in Antarctica. It's just amazing how versatile, how resilient they are, and then just their action, the way that they can really co-evolve with the bacteria that they infect too, and then manage to survive. They're amazing.
So so this picture that you have here, when I look at it, it looks like a spider to me, right? Like it has legs.
Well, I'm so glad you noticed that. This is a very common, this is what they call the lambda phage. And this is the lander, the typical lander physiology that you see. At the top here, let's see if I can use my thing. So at the top here is the capsid. So that's usually made out of different types of protein. And inside is the DNA. Now, one of the most cool things about the capsid head is that this DNA is packed in by a little motor that shoves it in, and so it's under pressure. There is a lot of pounds per square inch of pressure that is locking that DNA into that capsid head. And then what you've got here is a collar that kind of helps to attach the capsid head to the tail. And this is the tail. And at the bottom here, we have the base plate. Now, the base plate is frequently the site of recognition. So this is what actually attaches to the bacterial cell.
Yeah, that's what I was going to ask. If the little legs, if there's something specific about those legs, or, how is it that recognition happens? So you just answered that with that base plate.
Yes. Well, sometimes those legs, those are called tail fibers. And
Okay.
they will also recognize different types of receptors on the surface of a bacteria.
So does anyone ever study the proteins in those tails to understand what that protein-protein interaction is?
They do, yes. So one of my advisors, Dr. Docklin, does a lot with EM. So I use EM to help look at the structure of some of these phages. And they do look at the protein structures. They can do reconstructions, which I'll be learning in just a couple weeks. It's very exciting.
Yeah.
And once they can do that, then it helps them to figure out what those proteins look like.
Yeah, that's awesome. Other questions for Melissa?
Hi, Melissa, it's Effie. I was wondering, I love the history. I appreciate that. I knew a little bit about phages, but I had no idea why it was, bigger therapy in Georgia, in the country, right? So your history lesson was great. I appreciate that. My question is, how hard is it to isolate new phages?
That's a really great question. And the answer is that it's very complicated. So a quick outline of the steps to isolate a new phage is we start with what we call an environmental sample. So I get samples currently from the wastewater treatment plant here in Birmingham. And what happens is I take that wastewater and I split it up into some small flasks. And then I put a little bacteria in each flask. And then I grow it overnight. And then when I'm done, I strain it. And when I strain it, what I'm doing is removing all the bacteria. And what I hope to get is just the viruses in the liquid. So kind of like just old school Felix, Twart, all those guys, just get my little phage liquid. And then from there, it's the process of just testing it to see whether or not it'll infect certain strains of bacteria. And I work with pathogenic bacteria, so I'm really testing it against different strains of Pseudomonas and Staph aureus, a few strains of MRSA as well. And then once we find things that are actually effective to kill the bacteria off, then we have to go through a really intensive process of characterizing the virus itself. So to characterize the virus, we have to ensure that it's lytic only. Now, viruses are known to be really popular vectors for horizontal gene transfer. So you never, never want to use a lysogenic virus to treat some type of bacterial infection, because chances are what's going to happen is that virus is just going to end up transmitting antibiotic resistance between different types of bacteria. And so it's really important to really check and make sure that it's lytic only. And you can do that by looking at the plaques that form, but you also do that by conducting whole genome sequencing on the virus itself. And then once you've done the whole genome sequencing, you have to use either software or you can go through by hand and annotate the genome to make sure that it doesn't have any of the pathogenicity islands or anything like that that can be transferred from one bacteria to another, or that indicates like it has an integrase gene. So you know it's going to be able, if it has an integrase gene, to integrate its DNA into a host genome. Once you do that, then the process is really just, you get a lytic virus now, and so you're pretty sure it's only lytic. You know that it'll infect some stuff. At that point, then the process is really where you have to just amplify it. And then you amplify it and you have to go through some extensive purification. And at the point where you get it completely purified, then of course it has to go through a little bit of like animal testing before you would actually use it in humans. So that wasn't quite as quick of a summary as I had hoped, but.
That was quite long. That was great though.
Right.
Do you think that, have you looked at, when you talk about undergrad research, I was thinking this just hit me, at least the first part of this could definitely be developed into a cure or
you know, you're absolutely right. And I would love help doing that. You know?
I was thinking the first initial stages of just screening, you know, it's hard for me because I keep looking for, you know, some of these lab activities to do, but at a, and this sounds terrible, I don't mean it this way, but at a community college level, we're working with a population that hasn't had maybe the same rigors of science that we've had. And so maybe the first initial stages of, you know, collecting and being from a different area. I mean, I teach in Huntsville and Decatur, and Decatur has some nasty sewer problems that we could probably find some really good samples. So I thought at least the first characterization part, you know, or at least collection part, could be done through undergrad, you know, students, and then along the way develop talking about, you know, phage therapy as an alternative to antibiotics, which a lot of people don't even know that this exists.
No, it really hasn't, I think, hit the mainstream yet as far as the general population goes. I think people who really know about it are people who have sought to use it as a treatment for a loved one. And I would love to get your contact information and to work with you. You know,
Wonderful. I'm looking for an idea. So that sounds fabulous. And this is a topic that I think I teach a lot of pre-nursing students. You know, they take anatomy with me before they go into nursing. And I think it's good for them to have a better understanding of the possibilities that are out there. And I think it would probably blow their mind to even know that there is, you know, topical, you know, administration of this, you know, phage cocktail. I mean, I think they would just reading a paper on that alone would blow their minds. So
Excellent. Well, yes, I would very much like that.
awesome. I will email you. Thank you, Melissa.
Okay. And let me see, I see a couple more. Any common receptors or binding proteins in human and bacteria which can confuse phage to infect? That's a great question. I would say that there are not a lot of the areas that phage infect are either the pili or the flagella of a bacterium, or sometimes it is the pore proteins that allow the bacteria to help modulate the movement of different chemicals or compounds across the membrane. But I don't know for sure that that's the whole answer. So Anu, I will try to get a better answer and a more thorough answer for you and follow up. That's a really great question. And then let me see, I think, okay, user, I was wondering whether the phage therapy would work better with severe bacterial infection or minor infections. Well, now that has been really hard for them to assess, and that's because people with severe bacterial infections can frequently die, and it's not because of the phage that they were administered. It's frequently because of the fact that they just had a lot of complications, and by the time that phage is used as a treatment, it's like a last-ditch effort. And so I would say that as phage therapy becomes more mainstream in the United States, that it would be something that we would use as a first line of defense. And then you could use it very effectively with minor infections. And then when we find people who don't respond, patients who don't respond to that, then the severe bacterial infections would require us to do like a personalized phage cocktail. So we would have to take swabs and then produce something which would hopefully be as effective as treating a minor infection with just like a generalized phage cocktail. I like that question. And then from Luke, are bacteriophage generally more or less specific for particular bacteria than antibiotics? That is a really great question. So we have a different types of antibiotics, right? We have what we call broad spectrum antibiotics. Those are antibiotics that are effective against gram-positive and gram-negative bacteria. But then we also have antibiotics that are specific to treat either gram-negative or gram-positive bacteria. And I find that bacteriophage really are specific too. So what's unusual to see is a bacteriophage that infects gram-negative bacteria and also infects gram-positive. So phage have some similar limitations that we see in antibiotics. And if they have developed a phage that will infect across both gram-positive and gram-negative, please let me know, because I would love to read that article. Thank you for all the questions, guys.
This was really fantastic, Melissa. I learned a ton, and I'm sure everyone else did as well. So really appreciate the attendance from the audience too. And this is why we do this. So we all learn something new. And Melissa, thanks for all of the hard work and all the new knowledge that you're generating. I could see phage therapy really exploding as time goes by.
Yes, I really hope so. I love it.
You know, history has a way of doing that, right? So it circles back. And especially with all the multi antibiotic resistant bacteria out there, we need a solution.
Well, yes, never in my life would I say that there might be a silver lining to that terrible tragedy.
Right.
Yeah. Here it is. Maybe it's just phage therapy. It's causing us to think outside the box.
Right.
So it's
Well, thank
always
you
good
so
to-
much. Thank you for everything, and thanks for your attendance. Have a nice evening, everyone.
Everyone.
See you next time.
All right. Bye.
Bye.